Hello, this is Dr. Ellis going through the Year 12 exam. So let's go into it. First question, that's what I do on question one, is I would name them first before I do anything else. So let's look at number one. So I need to know which one is a monomer of cellulose. I've done the hard work. So yeah, it's a beta glucose, so it's D for that one. That's not hard. It's produced during photosynthesis and respiration. Well, if it's just photosynthesis, I could put glucose down. However, it says and respiration, so yeah, it is only C because respiration would use glucose. It can even use amino acids, can respiration. But yeah, if it's produced, it's the only thing that's a product. Forms a polymer that gives a positive result with a burette test. Well, that's the test for protein. Therefore, it must be B, because that's the amino acid. It's the only one that's got those groups that react with the burette test. And is a monomer of an enzyme's active site. Enzymes are proteins, so it's B again. Again, should have been an easy start. I can understand people getting confused between glucose, but yeah, look for those alternating OH groups. That's the sign of beta glucose. That's why each glucose then has to flip over, so you get that alternating glycosidic bond, and therefore straight fibers formed. Just move this up onto 1.2. I've done a bit of working out again, just to kind of show you. Yeah, so raffinose, never heard of this before. That's the aim. It's made of three monosaccharides. They all have the same formula. The number of oxygen, hydrogen, and carbon atoms. So I'm going to times C6H12O6 by 3. However, when I join a glucose to a glucose, it's a condensation reaction, therefore water is lost. So if I'm joining 3 together, I'm going to have 2 glycosidic bonds. So I need to remove 2 water molecules. So that's my multiplied by 3, but I need to knock off 4 hydrogens and 2 oxygens. Therefore, we get the fact that it's 16 oxygens. Hydrogen is 32, and carbon is 18. A lot to do for 1 mark. Number onto 1.3. Yeah, the biochemical test for reducing sugar. You'll get used to my umming and ahhing, or you'll turn it off because it will start annoying you. But let's see anyway. Yeah, so it's a biochemical test for reducing sugar gives a negative result. So I need to show the test for non-reducing sugar. So we need a new sample of it, and we need to boil a new sample. With acid, and then neutralize. That hydrolyzes the glycosidic bonds. So I go back to my monosaccharides now. So now I just need to do the Benedict's test. So then I just heat with Benedict's. Reagent, and then we get a color change. So it's describing the test. So it turns from blue, the color of Benedict's, to I'm going to say orange. There's a range that you can put. Semi-quantitative is the Benedict's test. I think people got this wrong on the exam. Again, Dr. Clark more this. It's the fact that you need to heat it twice. Yeah, I'm boiling it once, and then you then have to heat it again. You can't just it can't imply that you are still boiling it when you add the Benedict's reagent. Is the crucial bit. That's 1.3. Question 1 done already. I'm flying through. Oh, only 10 more questions to go. 2.1, explain the arrangement of phospholipids in the cell surface membrane. It doesn't say describe them, it says explain them. Yeah, so it forms a phospholipid bilayer. A phospholipid bilayer. As, in fact, I'm going to change that to, because. And that's what you need to think of in an explain question. Get to a because. Yeah. So water is found inside and outside the cell. Inside and outside the cell. And the phospholipid tails, I'm doing it in shorthand a bit, phospholipid fat acid tails are hydrophobic. Or I could say that the globular head is hydrophilic, and therefore they move away from water. Therefore, move away from water. Yeah, the cytoplasm's full of water. Tissue fluid is outside, that's full of water. So yeah, so I guess people get it wrong because they just describe it and don't say because. And the reason why it forms a bilayer is because those tails are trying to get away from water. 2.2. Describe how an ester bond is formed. So this is just knowledge. Yeah, so it forms, it's formed, right, so it's a condensation reaction. And it's between glycerol and fatty acids. Okay on there? Yeah, nice and straightforward. It should be easy. That one. And then the property of water that helps to prevent temperature increase in a cell. So yeah, it's the fact that water's got a high specific heat capacity. And yeah, it takes a lot of energy for it to change temperature. A lot of energy for water to change temperature. You know, like compare it to metal. Yeah, metal heats up really, really quickly, cools down quickly. Water heats up slowly, cools down slowly. And that's all down to the high specific heat capacity. I think Moxie said something about buffering changes. Yeah, so I'll probably go on to say it takes a lot of energy to change temperature of water. Therefore, organisms have time to adjust to temperature change, to adjust to temp change. So if I go into a sauna, it's like 50 degrees in the sauna. I don't suddenly become 50 degrees C. If I did, my enzymes would all denature massively quickly and I'd die. You know, I go into that sauna, I start to warm up slowly, and that gives time for my body to start to do things to cool me down. So I would start sweating, the sweat evaporates, and it keeps me cool or not the same temperature as the sauna. And it's the sweating that's supposed to be the good thing about a sauna. You're sweating out whatever's in the sweat. A sweat gland's like a mini kidney, so you will lose like impurities and things. Number 3, which is a bit of repetition, sorry, but yeah, describe how a phosphodiester bond is formed between two nucleotides. Yeah, so it's condensation reaction again. Condensation reaction, and it's between yeah, so it's the sugar and the phosphate. The trouble is I can't say sugar and I can't say, well, we allowed phosphate, but it's a phosphate ion. Yeah, but it's really between. So better answer with the phosphate ion and the sugar, but the sugar in DNA is deoxyribose. Deoxyribose. There we go. Get my 2 marks. This one's a hard one. I like to circle numbers. It's my technique. The 2 DNA strands, right? So it's a double-stranded piece of DNA. A particular gene contains 168 guanine bases between them. The relationship between the numbers of guanine bases and the 2 strands is shown by the following equation. Uses information and your understanding of the structure of DNA, so it's going to come into it as well, it's dual-stranded, calculate the maximum number of amino acids coded for by the gene. So let's substitute the numbers in first of all. I've got 168 guanines equals 4 lots of A plus T minus C. Well, I know what C is. C is 168, because for every guanine, there's a cytosine. He then reaches for his calculator. Ugh. I'm not going to do it. I should be able to do this for a head, but it's Saturday morning, so I'm going to risk it. Right, so I'm now going to add 168 to 168. So I've got 336 equals 4 lots of A plus T. Let's divide by 4 to get what A and T is. Divide by 4, which is 84. 84 equals A plus T. Therefore, A equals, well, half of that, which is 42, and T equals 42. Oh, so now I know everything. I know A, T, G, and C, and I know I've got 42 plus 42 plus 168 plus 168 of them. So let's add all that together. So I've got 84 plus 168 plus 168, which equals 420. So I've got 420 bases. Let's scroll up slightly. Run out of space. Which equals 420 bases. That's my gene. 3 bases code for 1 amino acid. 3 bases code for 1 amino acid. So therefore 420 divided by 3 is 140, which equals 140. However, I've gone wrong there, haven't I? Let's because I've not used my DNA structure. I used my A, T's, G's, and C's. But yeah, that's 420 bases over 2 strands. On 2 strands. Therefore, it's 210 on 1 strand. So it's 210 divided by 3, which is 70. Yeah, 70 amino acids. 70 times 3 is 210. So yeah, a lot to do for the 2 marks. Well done if you got that, because there's again you have to be comfortable with maths, a bit of rearranging. No DNA is double-stranded, and that 3 bases code for 1 amino acid. So there's lots to do there. And think you move from GCSE where you get 5 marks for basically doing pi r squared, and now it's like, yeah, there's lots to do, and I'm only getting 2 marks, and it's like, oof, that's a bit mean, but it's welcome to A level. 3.3 is just histone. Protein is a histone, and semi-conservative DNA replication. The 2 strands within a DNA molecule are separated. Each acts as a template for the formation of a new one. Describe how the separation of strands occur. Names of enzymes, names of bonds. Yeah, so DNA helicase breaks, not hydrolyzes, breaks H bonds. I would write hydrogen in the exam, between complementary bases. Yeah, names of enzyme, names of bonds. Yeah, it's not hydrolyzing because it's not a chemical reaction. DNA helicase is spinning. It needs ATP, and it spins round, and it physically just pushes the bonds, the strands apart. Hydrogen bonds are just like a little attraction between a positive and negative area, and therefore, yeah, it can be broken just by pushing by helicase. That's question 3 done. I'm romping through. We are hopefully you're still with me. Let's have a look at 4. This is a bit of terminology. Yeah, describe how a phagocyte destroys a pathogen present in the blood. I know a lot of your answers were like more detailed about this, but yeah, I mean they allow one. Just to say, you say they engulf and digest Yeah, and you get 2 marks for that at GCSE. So you still get 1 for engulf. So it engulfs the pathogen, and it forming a, it's called a phagosome. Phagosome. Or I could say a vesicle that contains the pathogen. Vesicle with pathogen. So you don't need to know phagosome. I just think it's easy to know it than like vesicle containing a pathogen. And then we need to get the fact that, and then this fuses with a lysosome, which fuses or joins with a lysosome. So engulf gets a mark. All this gets 1 mark as well. And then the last bit is the enzymes. So I'll be saying, yeah, the lysozymes consist of enzymes that digest the pathogen. Super. 4.2. Yeah, for next, where antibody-antigen complexes form. Again, it's nice and easy on the ends here, not on the end here. This is where you'd attach things like a dye molecule or an enzyme or a drug if it was monoclonal antibody technology. And 4.3, what's the role of the disulfide bridge in forming the quaternary structure? I just say literally what it does. So it is literally holding 2 polypeptide chains together. Yeah, so it joins 2 polypeptides together. Do I get the marks there? Joins 2. I do. Yeah, it's only worth 1 mark. And just see how many lines is it? 1, 2, 3, 4, 5 lines. Again, years ago they used to give just 2 lines for that in case you got it wrong, and then you had a spare line to write it. But they were found that people were asking for extra paper in the exams, so then they put 2 lines and then they'd say extra space and put 2 lines. However, that was messy. So now they just give 1, 2, 3, 4, 5 lines for like a 1 mark answer. Yeah, again, you're not expected to fill the space. If it's 1 mark, you should be able to answer it on 1 line. So yeah, it's more expensive for the exam boards to deal with extra paper. So they've massively increased the number of lines that they give. Right, 5.1. Eukaryotic cells produce and release proteins. So we're in a eukaryote. The role of organelles in the production, transport, and release of proteins from the eukaryotic cell, and no need to talk about transcription translation. So I'm just going to start with the REI is the first. Yeah. So So what's a pen gone lighter? Who knows? REI makes proteins by its ribosomes. Again, I was going to write about mRNA, but it says don't include detailed transcription. Then we have then vesicle forms. Vesicles form and transport proteins to the Golgi. No mark for Golgi, it has to be apparatus. And then the Golgi apparatus modifies and packages proteins into more vesicles. And then these vesicles fuse. So the word again, fused, as in join with. the cell membrane to release the proteins outside. And it will be proteins. You won't get a protein in the vesicle. There'll be thousands. Release the proteins outside the cell. It's got a name, so we may as well we'll put it. Yeah, this is exocytosis. Exocytosis. Have I got the marks? Let's look at the mark scheme. Mark point one, DNA in nucleus is code for protein. I didn't put that one. Number two, ribosomes or the rough endoplasmic reticulum produce proteins except REI. So I've got that one. That's good. Golgi apparatus modifies proteins. I've got that one. Good. Vesicle transport gets a mark. That's good. And vesicle fusing with cell membrane exocytosis. Good. What I've missed is DNA nucleus is the code for the protein. Don't know. I would never have thought of putting that. However, you can always add as well. Yeah, that ATP from respiration is needed for this. ATP, from respiration is required. Yeah, it's needed for protein synthesis. So to load an amino acid onto a tRNA, that needs ATP. To form a vesicle and move a vesicle, that also needs ATP. So vesicle movement also does as well. So I could have got another mark there, but I've still got my 4 marks. My credibility is still there. I've not dropped a mark yet. Oh, close though. Number 5. Relatively straightforward. You just make sure you know that a plasma cell, right, well, that's a white blood cell. That's a type of lymphocyte. So it's a eukaryotic cell. So where would I find a nucleus? Well, not in a prokaryotic cell, but a leaf in a plasma cell do, so it must be B. So the intersection there, and cell wall, well, plasma cell hasn't got a cell wall, it's an animal cell, but leaf cells do, and bacteria do, and they're prokaryotes, so it's D. And 1 for each. So yeah, that seems more generous than some before, where we had lots to do for the mark. Right, I'm going to struggle to do this one because I haven't got the actual size of the thing, so I can just go through the method. The key thing on any magnification is just measure the scale bar and measure it in millimeters. I think I've got I don't have the actual value, but let's pretend I have measured it and it's 30 millimeters long, 3 centimeters. So so my image equals 30 millimeters, which equals 30,000 micrometers. Get it into the same unit, and then after the magnification. Well, the magnification has no units, so I must be dividing 70 and 30,000, and the magnification is going to be large, so it must be the big number on top. So 30,000 micrometers divided by 70 micrometers, they cancel. And 30,000 divided by 70 equals 429. It's 2 significant figures, not 2 decimal places. I think the answer that was 460. So 30 something, it'd be a 30 and a bit millimeters. 5.3 is a suggest question. Why is the nucleus not visible? Well, it's a slice through the cell. It's a section, so it could be somewhere else in the cell. So I'll be putting, the cell is 3D. This is a 2D image of a section. Therefore, the nucleus is elsewhere in the cell. It's not in this area. Organelle S, well, that's our vacuole. T is a chloroplast. Do I need permanent vacuole? No, I don't. They're happy with vacuole. That's good. And explain the advantage of using a transmission electron microscope over an optical microscope. So it's got higher resolution. but I have to explain it. Higher resolution, because now I can't say it here because it sounds like I'm talking about the microscope, because electrons have a short, no mic, get a mic, wavelength. Good. It's only worth 1 mark, so I'm happy with that. 6.1. The mechanism that causes lungs to fill with air. Right, so I need to raise my rib cage and lower my diaphragm. So I need to get this the right way around. So it's going to, I need to know it's the external intercostals. Intercostals contract, and the diaphragm, I said it as I was going to spell it and then spelled it wrong. Diaphragm contracts. So that causes the rib cage to move up and out, and diaphragm to lower. And this increases. I'm just going to put an up arrow just for speed, but I would write increase. This increases the volume of the thorax or the thoracic cavity. Therefore, and lowers the pressure. Lowers the pressure. So then, air enters the lungs down a pressure gradient. Stroke, moves in to equilibrate pressure. That's how we've done. Diaphragm muscle contracts and external intercostal muscles contract. Yes, so we've got the first mark. Good. Causes volume and pressure to decrease. Causes volume to increase, pressure to decrease. Yes, I put that. So there's no mark for a second bullet point. That's annoying. And then marking point. It's only a 3 marking tape. I've wrote 4 anyway. Air moves down a pressure gradient, or air enters from higher. So I've got that as well. But it's good practice, I think. If it's worth 3 marks, write 4 things. See how I'm putting bullet points here? Yeah, good practice, I think, because it makes me think of like different points that I'm trying to say to get all 3 marks. Can I bullet point it in the exam? Absolutely. Are they a dream to mark? Yes, they are. What people tend to get wrong, or do is that they'll say the mechanism that causes the lungs to fill with air is, so they repeat the question when there's no need. Just give the answer. Then we've got this micrograph of lung tissue. K is an air-filled tube and L is a blood vessel. So it's told me it's an air-filled tube. So that's going to be, K is going to be a bronchiole. It's a small tube because it's right next to an alveolus. See the alveolus here. So it's not the bronchus, the bronchiole or the trachea. And L's a blood vessel. However, it's got a thick wall. So it's going to be I'm going to guess it looks like more like an arteriole to me. Arteriole. I could put artery or vein. I just can't put capillary. It's not a capillary because it's got a thick wall. As you know from GCSE, they have a 1 cell thick layer. Number 7. A molecule of tRNA is made from a precursor molecule that is modified, and that's on the left. Modification includes splicing, right? I'm going to underline a bit of this. Splicing, trimming, and attachment. of new nucleotides. Diagram shows the precursor for tRNA for the specific amino acid proline, and a tRNA molecule specific for the amino acid proline. Some of the bases are shown in the diagram. Yeah. So let's have a go. Which bond is labelled Z? Well, that's the as you can see here, it's the bond between these. This is a chain of mRNA. Sorry, of RNA. It's not mRNA. So yeah, so it must be hydrogen bonds. That's our hydrogen. And describe how the precursor molecule is modified to produce a tRNA molecule with specific for this amino acid proline. Well, I've lost. That's gone, and that's gone, and it looks a bit like that's gone as well. So the shaded areas are removed. Okay. So yeah, nothing like groundbreakingly difficult. Yeah, so I'm going to say it's there's removal of these shaded areas. I think they're introns. So that's what it said in my question. They undergo splicing. Removal of the shaded areas, and if they've been removed, you'd be like you would hydrolyzing phosphodiester bonds to remove them and then forming them to join them back together again. What else has changed? Well, this is new over here. So there's an OH appeared and an ACC. So I can add that as well. So 3 nucleotides. With bases ACC are added. That looks like the amino acid binding site. This is because this is the anticodon there. That's right, anticodon. This is the amino acid binding site. What else can I say? Yeah, my anticodon. So the GGU has moved to the anticodon position. So I'm going to put that as well. GGU has moved to the end to act as an anticodon. Brilliant. Let's have a look at my mark scheme. How have we done? So description that references the following: removal of the shaded nucleotides or introns. Yes, I've got that one. That's good. Attachment of ACC. Yeah, get that one. And the correct reference to phosphodiester bond. Yes, I've done that as well. And there's no mention of the GGU moving. That's annoying, but that's mark schemes for you. Again, it's 3 marks. I've put 4 bullet points down, and you do play guesswork. What's going to be on the exam? Sorry, what's going to be on the mark scheme, and you play the guesswork. What's on the exam as well. But yeah, that's not that surprising that I've not got 4 marks on a 3 mark question. Right, on to 8.1, the controversial question, because it shouldn't have been on the paper. So this is the one that was discounted or counted if it helped or hindered your score each way. But it is just the it's the required practical. It's the only bit where it's the only required practical where we actually need to know the actual method that you did. So crucially here, you need to use the it says root tissue. You need the tip. So I'm gonna take 2 millimeters of the root tip and place in a staining block. That's my first one. Then I'm going to add it was hydrochloric acid, and we also used acetic acid to soften the tissue. So yeah, it stayed in there for a while. Then we popped onto a slide. Yeah, so place tip, slide, and tap with a glass rod. I've done this out of order here to separate cells. But I'm going to recover. This is done after a drop of stain is added. It needs to be stained, not dye. Then we add a cover slip. Firmly with thumb to squash. It's called a root tip squash. To squash the tissue and gain a single layer of cells. There we go. 4 marks, easily. I don't know, I put up enough for 5 there. 8.2 shows the cell cycle. Interphase is divided into 3 parts. Where's the S phase? So, well, the S phase showing the amount of DNA in the cell. So, in the S phase is when DNA replicates. So, the start is there and the end is there. Yeah, it's that diagonal up bit. And then 200 cells were counted, and the mitotic index was 12%. So remember that's so mitosis, number of cells divided by total, times by 100 equals 12. However, I want those that are in interphase, so that's the rest of them. So if 12% are in interphase, that means that 100 minus 12 equals 88% in interphase. 200 multiplied by 88% equals 0.88 times 200 equals 176. So yeah, relatively easy. Most people got 0.2 and 0.3 right. Right, hard one. Bit of maths again. A box is drawn around one of the cells shown above. The box width is 8 plus or minus 1 micrometer, and the length is 10 plus or minus 1 micrometer. Calculate the area of the box and calculate the absolute uncertainty. So let's get the easy mark first. So area equals 8 multiplied by 10, which equals 80. And that would be in micrometers squared, because I'm doing micrometers times micrometers. So that's 1 mark. Let's do the uncert equals, well, that's 1 over 8 plus 1 over 10, which equals, so 1 divided by 8 equals 0.125 plus 0.1, which equals 0.225. So that's my uncertainty. Or I could say that's 22.5%. Then I apply this uncertainty to my answer. So 80 multiplied by 0.225 equals, well, this is going to be my uncertainty, multiplied by 80, which equals 18. So that's called the absolute uncertainty. So my answer is 80 plus or minus, see how it's written above here, plus or minus 18 bracket micrometers squared. Again, the number of people who didn't put squared on the micrometers and didn't get that 1 mark. So well done if you got all 3. if you get plus or minus 17, and quite a few people did, I've no idea what you've done wrong, but yeah, it's not 18, therefore you don't get the mark. So yeah, always get the easy mark by just working out the area, and then do, as you can see here, it's the what percentage is the uncertainty for that one, what is it for that one, and then you add them together. So there's one mark, even if you get it all wrong, for adding together your uncertainties, and then I apply that uncertainty to my final answer of volume, so area, and therefore, yeah, that's my uncertainty now is 18. So yeah, it's knowing the rule. When you're multiplying up values, you add the uncertainties. Yeah, and then this is, yeah, that's metaphase, and that's anaphase. Should be easy. I think that was the easiest one. The process that takes place at the end of telophase, well, that's cytokinesis. Cytokinesis. I got a few exocytoses there on that one. And then this one that's, again, you need to transition to an A-level description. So it's a describe question, not an explain question. So it's the effect of this agyl concentration. So as the concentration increases, I can't say that the mitotic index decreases. So let's have a look at these numbers. So I've got a decrease there, that's a decrease, that's a decrease. However, that's a very slight increase. And that's decreased loads. That's decreased by 2, and that's decreased by 1, and then there's only a slight increase. So that's what I need to describe in my answer. Yeah, so as agyl, again, I would put it increases the mitotic index, mitotic. I can't spell mitoxic. Try again. The mitotic index decreases until 1.5 ppm of agyl. Yeah, I need that. Yeah, when it stays roughly constant. It's only changing by 0.1. It's increasing by 0.1. Constant. I think I need a break. And then I need to get this rate of change. If I plotted this data, it'd be a graph that looks like that. Yeah, so I need to say, yeah, that there's a large increase. Sorry, large decrease in mitotic index, MI, with the first 0.5 ppm of agyl. Or it's, yeah, there's different ways of saying it. What most people did, they give a very brief description and then went into an explanation. And filled all the space. However, it's just a describe question. So if you're disappointed with your mark or you want to make up marks in the future, work on this. It's exam technique. It's making sure that you say the right things for the question. And then 8.8, chemotherapy as you treat cancer, involves uncontrolled cell division. This chemical I never heard of is a chemical used to preventing shortening of spindle fibers. Preventing shortening of spindle fibers. How it affects mitosis, right? So that means that if it prevents shortening of spindle, well, I'm going to say what spindle does first. So yeah, spindle attaches to the centromeres of each chromatid of a chromosome. So yeah, so the centromeres can't split. So yeah, chromosomes can't move to opposite pole. Can't move to the opposite poles. The cell is stuck in metaphase, so can't carry out anaphase. So yeah, so those are our marks. Again, marks for knowing that the spindle attaches to centromeres. So centromeres can't split, so chromosomes can't move to opposite poles, and then yeah, it's stuck in anaphase. There was a mark therefore that the cells produced have incorrect numbers of chromosomes. Chromosomes therefore can't divide it in the future. Super. Right, to question 9. I'm ploughing on. I'm fading, but I'm ploughing on. Let's go for it. Number 9. So investigate the effective temperature. That's our independent variable on the hydrolysis of lipids in milk. So I'm doing a lipid thing. So add 5 centimeters cubed of milk, and that should be a small c, sorry. 7 centimeters cubed of sodium carbonate. Those are both control variables. 5 drops, and that's another CV, control variable. So I'm measuring pH. The pink colour, put it in a test tube at 30 degrees C. That's a CV as well. After 5 minutes, add 1 centimeter cubed of lipase. That's another CV. And start timing immediately. Stir the contents. Record the time taken for the pink colour. Yeah, so that's our DV. The time taken for a pH to change. Repeat it. Experiment at 3 other temperatures. Oops, have I just put CV on the temperature? Oops, idiot. As in me. Yeah, so that's our IV, isn't it? That's one of our independent variables. The methods, oh yeah, and it tells us that phenolphthalein turns a pink colour in alkaline conditions but becomes colourless in a solution with a pH below 8.2. Right. So an acid must be being produced that's making the pH change, that's causing the colour to go. The method states how some our variables are going to state, give 2 other variables the student should control in this investigation. So I can't put the ones I've already put. Okay. However, it's talking about volume of lipase. We haven't controlled concentration of lipase, so that's a good one. So yeah, concentration of lipase. It says stir the contents. So yeah, how much stirring? I could always put. So the so how much to stir? There we go. Let's just check I've got them right, because concentration. Yeah, so I could say concentration of all of the things. Speed of stirring. How much to stir? I'd probably get that, I think. Yeah, but I maybe should put the frequency of stirring. They also allow the same person to judge this when the solution loses pink. Yeah, it's quite subjective, is that? So, and people have different opinions, so that would also be valid. Normally, you don't get a mark for the same person, but if it's like a visual colour change, then yeah, appreciating that different people have different opinions is valid. Give one reason why the student put the test tube in a water bath for 5 minutes before adding the lipase. It's so that the reactants are at the same temperature. So yeah, reactants are all at the desired temperature. That's good. And in this investigation, the student did not add a buffer. Well, buffers control pH. So I don't get a mark for saying buffers control pH. Well, I'm measuring pH change. Yeah, so that's what I say is that we are measuring pH, comma, semicolon, sorry. A buffer would stop this. I should put change after that. I did say it, but then didn't write it. All these answers are really easy. Yeah. However, nationally, when these questions are done, it's like, half marks is kind of A grade standard on these. So hopefully, as we go through this, you're thinking, oh, God, oh, that's easy. All right, yeah, that's actually quite obvious. So, and it's knowing that when you get to these like practically based questions, just try and think, well, put the obvious answer down. 9.4, describe how the student would have maintained the temperature during the experiment. So you don't get a mark for just use a water bath. So it's knowing that, in a water bath, I'm going to draw this out. I'm not going to write it. So there's a beaker. There's my water. In here goes a boiling tube or a test tube, and the thermometer is inside there. And there's my kind of like solution that's inside my boiling tube. So so this is my water at 30 degrees C. That's my thermometer, which we check at regular times, and when it cools down, if less than 30 degrees C, add warm water to beaker. So you need to say that I'm going to place the boiling tube with a test tube in it in a beaker of water. The beaker's got water at 30 degrees C. And then throughout the experiment, if the temperature drops in the boiling tube by checking the thermometer, you're going to add some more warm water to the beaker. Or you could have had a very hot one here. I could have a water at 60 degrees C and maneuver the boiling tube forwards and backwards to maintain it at 30. That's another method of doing it. It's probably what we did because it's easier to do than start like boiling kettles when there's a class for like 20 students who all need a kettle boiling. It just makes it manageable. 9.6, students' results are here. So we've got our time taken to the pink colour to disappear. And read the axis. So as I increase temperature, the time taken is getting less, so it's getting faster. And then at 40 degrees C was my lowest temperature, lowest time, fastest speed. It goes up. It gets slower again up to 50. Student concludes that 40 degrees C is the optimum temperature. Well, it could be anywhere between 30 and 50, wouldn't it? It could have done that. That's badly drawn. I should do that again, sorry. Could have done that, and therefore the lowest temperature could be here. Or it could have done that, and the lowest temperature could have been there. So either of them. So I've only got 10 degrees C increments. So I've only got, only gained data for 10 degrees C increments. Optimum could be between, I'm getting faster here, 30 and 50 degrees C. It could be anywhere. Then we're on to a good old explain question. Explain the effect of temperature on lipase activity shown by the graph. So why does it get faster as it gets warmer? So so the rate increases of reaction goes up due to more Ke, therefore more ES complexes and collisions with required activation energy. The more kinetic energy I have, the more bumps we have together, more enzyme subject complexes form, and the more likely to have the required activation energy. And then the rate goes down as more enzymes are denaturing. They all don't just denature at a temperature. The chance of denaturing increases with temperature. Therefore, fewer. to form ES complexes. It's a nice easy one. Then we've got a tangent question. This will be a challenge on the iPad. Again, I'm just going to go through, I'm not going to do the working out here, but yeah, it's the rate at 60. So I need a line there. So have I got a line tool on this? Not really. I have to draw it. Anyway, so, I'm drawing a tangent, kind of zooming along there. Again, it's not the best tangent ever. It's awful, but you get the idea. Extend it to the axis and extend it to the axis, and make sure you read off. So that's gone up. This one has gone up 1, 2, 3, 4, 5.2. So my y distance is 5.2, and my x distance is, oh god, that's an awkward one. I keep saying God, sorry, not normally a blasphemer. But yeah, let's assume I've gone to there, and therefore it's 90, 100, 110, 120. It's really awkward scale, isn't it? Anyway, I get the idea. Yeah, I'm not going to spend ages umming and ahhing over my value. So yeah, it's y over x, and just make sure that you're cutting on that scale's not starting at 0; it's starting at 3. So yeah, be careful on that. And explain the change in pH shown by the graph. So yeah, why did it decrease quickly at the start, and then why is it leveling off? Yeah, well, this is one experiment. Therefore, there's lots of collisions at the start because there's a high substrate concentration. So we've got at the start, or I can say high initial rate, has lots of substrate, therefore lots of collisions where fatty acids are produced or made that lower pH. Lipids become limited, lipids run out, therefore the rate slows and stops. I.e., there's no substrate left. I've done that 9.8. Yeah, it decreases as fatty acids are produced, so I've got that one. High initial rate because there's lots of substrate. High ES, i.e., high chance of ES complex. Yeah, and then it runs out. Got my 3 marks. Number 10. Suggest what stimulates the spiracles to open. So this is scientists measure the change in concentration of oxygen and CO2 inside the tracheoles of an insect. So it's in these tubes inside the insect. Suggest what stimulates the spiracles to open. See, I've got a graph here with a numbered scale, so I'm going to use it in my answer. And I'm making sense of it first. So I'm in a tracheole inside an insect. So and the insect's using oxygen. So here I've got like the oxygen's increasing. Well, that's when the spiracles must open. So I'm just going to add that to here. Yeah, so that's where spiracle opens. So S open. And then that's when it closes. Spiracle closes. Because that's where the oxygen goes down. So the thing that's causing it, see how I've got an increase here. Let me highlight a pen. So it's increasing. So it can't be oxygen because it's not doing anything. You can't just like, oh, it does nothing and then it suddenly opens. It must be this increasing in the carbon in the carbon dioxide that's causing it. Yeah, so that's what I'm going to say in my answer. Yeah, it's the increase in CO2 to, and then so let's take our value from it. So it's 2.8 arbitrary units. It's the increase in CO2 to 2.8. Yeah, that causes opening. Let's see what the marks you said, because I think, is that 2 marks? Is that 1 mark? Yeah, so increasing carbon dioxide concentration, 1 mark. Correct reference to 2.8 arbitrary units. Good, I've got 2 marks. Super. On 1 line as well. I've done well. Give the total length of time, right? Because there's 2 of these on here. That the insect spiracles were open during the period shown in the graph. Explain how you arrived at your answer. So yeah, so I've already highlighted them here. So where's my highlighter pen? So it's open at 4 seconds and then it closes at 6. And it does the same over there as well. Ugh. So I've got 2 seconds there and 2 seconds there. So I've got 4 seconds worth. Yeah, so total amount of time is 4 seconds. From right there. Yep. And the reason why it's that is because oxygen concentration increases. Yeah, O2 increases as it diffuses from outside and diffuses into tracheoles from the atmospheric air outside the insect. Good. And then large insects can contract the muscles of their abdomen to force air in and out. The spiracles, called abdominal pumping, increases gas exchange efficiency. Give reasons why large active insects. Right, so I've got large active insects use abdominal, while small, less active insects do not. So I've got large v small, active versus inactive. So let's do the large first. So large insects have a smaller surface area to volume ratio. Therefore, by pumping, they are increasing the it's like the concentration difference, i.e. it helps maintain a concentration gradient for O2 diffusion. So it's like us breathing. When I breathe, I put fresh air into my alveoli, and therefore, every time I do that, I keep the diffusion gradient going. And I'm saying that by being a large insect, it needs to do this more. So it pumps its abdomen because it's a bigger insect and it's to battle that concentration gradient more. Active, so yeah, if active, it requires more oxygen for aerobic respiration. And then it's the same answer again, really. So let's see how I've done. So large insects have a higher metabolic rate, stroke rate of respiration, so higher demand for oxygen. Yep, I've said that. It's active, it requires more oxygen for aerobic respiration. Large insects have a longer diffusion distance to tissues or have a smaller surface area to volume ratio. Yep, said that. Abdominal pumping draws oxygen-rich air in due to a decrease in pressure, or it forces carbon dioxide out, or pump air in and out, so maintains. Yeah, sorry, I've got it. Maintaining a concentration gradient. Phew. And then we're getting there. 10.1. Explain how HIV affects the production of antibodies when age develops in a person. Got to be gone for more than an hour. HIV, yeah, well, HIV kills T helper cells, therefore there's less cytokines released to activate B cells. So then I've got fewer B cells to clone into plasma cells, but during mitosis. So therefore, so then I've got fewer plasma cells to make antibodies. There you go, answer the question. I've got antibodies mentioned at the end, good. I've got there, and I've got 3 ways of getting there. Yeah, so it kills the cell, less activation, fewer plasma cells, and then that's my answer. So yeah, so my 4th bullet point isn't needed, but it's there just in case it was a mark. Then over here, the scientists measured the effect of a drug on the number of T cells and the number of HIV particles in blood taken from a person with AIDS. The results are shown in the graph. This is where again I get my highlighter pen out, and I'm going to go over the line. So this is T cells. This line here is showing T cells. So that's this axis here. So these numbers here are on my green side. And then I've got my HIV particles here, which are going up and down, and that's on this axis here. Yeah, so that's that one, and that's the green one. Good. And synthesis of AIDS occurs when a T cell is below 200. So let's pop that on, a line on there as well. So that's our critical line here, across here. Yeah, so that's when my green line's below there, it's going to be yeah, so there's like a danger zone here, isn't there? Use all the information, all of this information. Yeah, so it's right. So it's from a person with AIDS. So this is only one person. To evaluate pros and cons, the effectiveness of the drug treatment in treating AIDS. 5 marks. So I need to give 5 observations here. So let's go for why it's good first of all. Yeah, so I'm going to give a plus. Yeah, plus is a pro about why it's good. So there's only there's only a short amount of time, short period, when AIDS is not treated. Yeah, approximately 4 months. Another one is that T cells are increasing. Increase 4 months. That's good. Let's go for a negative. There's a decrease in T cells. There's a decrease in T cells from 2 to 4 months. That's not good. We've only got one person. Yeah, only one person. Other people might respond differently. Other people may respond differently. In terms of treating AIDS, I'm never getting rid of HIV. Yeah, so HIV never becomes 0. Therefore, infection continues. And HIV numbers are higher after 16 months. HIV numbers are higher after 16 months. We've only got data for 16 months. Don't know the effect afterwards. Okay, so it's just been like that. I mean, I've put 1, 2, 3, 4, 5, 6. I've put 7 observations here. It's a 5-mark question. Yeah, I can carry on doing this as well. I just keep putting observations down. But the good thing I did, I've made sense of the graph. I put my 200 line on. I've found out the area where it's a problem. I've said about my green line, and I've said stuff about the red line, and I've gone to the text as well, and I've said stuff about that. You could also say that there's no stats test performed, but we haven't done stats tests yet. But that's how I would encourage you to answer those questions, because there are lots of potential answers, and I'd easily get 5 marks on that question. And they're tricky. Last question. Describe how the structure of glycogen is related to its function. So I need 5 points here where the structure relates to function. So let's do my first one. Well, glycogen, I was thinking it looks like that. It's a branchy molecule, isn't it? So this is like a zoomed out picture of glycogen. Oh, so let's get the first one done then. So yeah, so by being kind of like curly and stuff, it's compact. So yes, it's a compact molecule. Therefore, lots of glucose stored in a small space. I've mentioned glucose, so it's made of glucose. I should do it later, actually. It's also highly branched. Therefore, quickly hydrolyzed. Hydrolyzed back into glucose. Glucose. Yeah, I'm going to elaborate here as well. As animals have varied energy demands, glucose is made available quickly. Quickly to fuel respiration. Not sure if I'll get a mark for it. I'm just trying to think how on earth I'm going to get 5 marks out of this question. So that's that one. It's also insoluble. Therefore, lots can be stored. Lots can be stored inside cells without affecting the water potential. Therefore, no osmotic effect. Scratching my head a bit now, must admit. So I'm going to have to look at the mark scheme. Let's see how I'm doing. Helix called branched, so compact. I've got that one. Branched, therefore, also, yeah, compact. Compact molecule. Yeah. Oh, have I got the mark? Helix called branched, so compact. Right, well, I've got it. A compact molecule. Yes, yeah, so I've not said why it's compact. Due to branching. So I need to put here, yeah, due to branching. Due to its branching. Now I've got the mark. Polymer of glucose, so easily hydrolyzed. I haven't got that one. Branch, so more ends for faster hydrolysis. Right, I've got that one. Highly branched, quickly hydrolyzed. So provides respiratory substrate. I've mentioned respiration. And insoluble, so not easily lost from cell. Insoluble, therefore, doesn't affect water potential osmosis. Yeah, I've got that one as well. So I've kind of failed on number 1. I've got 3 there, but I've certainly got 3 out of 4 out of 5. So the other one is that a polymer of glucose, so easily hydrolyzed. So this I guess is knowing that we have enzymes to break down glycosidic bonds. So I think that's what it really means here in terms of this more scheme. Yeah. So it's yeah. So um, yeah, glycosidic bonds are easily hydrolyzed to release glucose. I think that's the marking point I was missing. Just the mark scheme sounds a bit strange on that one. Anyway, I've got through. Hopefully we found something useful there. Again, I've been going for a little while, so I will stop and have a celebratory glass of Vimto.